tour in a taxi this morning through Kiev, 1 hour. The cab driver was not able to find the exposition center. Anyway, here I am. Thank you that you're here as well. I must apologize for the late start. But now, welcome to the second day of the BeeBook session here. I'm happy to announce that the majority of speakers is actually here, so we can actually do that. I would like to remind the speakers to please try to keep a bit of time, but let's see how long we can make it to the break so that we can coordinate with the other groups. So the first speaker, I would like to ask, sorry, I may pronounce it wrong, Khem from Nepal, on nesting site preference and behavior of the giant honeybee Apis dorsata, please.
Good morning. Good morning, everybody, and namaste from Nepal. I am from Nepal. My name is Neupane, and the topics for presentation in this second session of this biology, my topic is the nesting site preference and behavior of giant honeybee Apis dorsata. Nepal, one of the small countries in the whole world, is very rich in honeybee diversities. We have different species of the honeybees. The Alexios-Leandros, that is called the rock bee, is the biggest honeybee in the world, which is found more than 1,500 meters from the sea level, that you can see here. You might have heard about the honey hunting of these bees. And second largest bee of the world is the Apis dorsata, which is found below 1,000 meters. And we have those bees also in Nepal. And this is the Apis dorsata honeybees. And third, our native bee is Apis serena. This Apis serena honeybees is domesticated as well as wild in condition in Nepal. And beside this, we have Apis florea, the little bee, and this is also still wild, and it is found in the bushes in between the lower mid-hills of Nepal. And one more, that is the stingless bee, Trigona. This is also found in the wild, in the forest of Nepal. And this is Apis mellifera, as you have in Europe, is the exotic bee for us. And the commercial honey production from this Apis mellifera is getting up in Nepal. Now, this Apis dorsata, they build a single large comb, and it is the open-nested honeybees. And in this single comb, they put the honey and bee bread at one of the corner of the comb and produce brood below this. And they build their nest in trees, in buildings, and live family together. And they live in one place, rear brood, and stay there until the season is favorable, until the food is available for them. And this is this bees is one of the source of good income for the rural people in Nepal. And the role of Dorsata bees for pollination is very high in Nepal. The distribution of this Dorsata bees in Nepal, this is a figure of Nepal, and this Yeah. The Dorsata Dorsata bees, they are distributed from the west to the east to the lower part of the Nepal. And then they have never been found above 1,000 meters from the sea level. And methodology done to find these findings. All possible nesting sites of Dorsata were searched within these districts, and the number of nest studies were about 500 in these districts, and out of these 500 nest, live nest as well as the old nest, 6 nests from the buildings, residential buildings, and 6 nests from the trees and 6 nests from the water tower were selected for these studies. And the size and the shape of the nest and cells of the worker, queen and drone bees were studied, and also the nesting site preference of the bees to buildings, trees were also studied, and also the color, texture of the sites they prefer were also the studies during this time. And the highest preference was given to the water tower. As you see here, these are the water towers where they give the highest preference to build the nest, and then to the residential buildings. And least preference was given to the trees. Even there were big trees with big horizontal branches of this Bombay species. They gave preference to the buildings rather than the trees to build their nest. And the size and shape of the color varies as per the growth of the comb took place. At the beginning, the shape of the comb was round or oblong at the arrival time. Then gradually it became elliptical when it was fully grown. And the smallest size of the colony was 10 into 2 into 5 cm when arrived at the nesting site, and largest was 130 by 30 by 60 cm as length, breadth and width, especially when it was fully developed in April month. The growth of the comb took place both in height and length. Apis dorsata colonies can live together freely, sometimes even touching to each other. You can see there are 2 colonies of Dorsata in three different colonies of the Dorsata, and 2 colonies they are about to merge, and here you can see the 3 colonies almost touching to each other here. That means it indicates that this Apis dorsata colony can live freely together. And this is one of the interesting subjects in honeybee biology, because when if you mix the 2 queen-right colonies in Mellifera serena, they will fight against each other and many bees will be killed. But in case of Dorsata, it's very interesting because even by they are finally together, touching to each other, 3 queen-right colonies. Findings: when Apis dorsata discontinued to rebuild their nest on the previous year's nest, because honeybee, they gave the highest preference to build their nest on the previous year's nest, but they discontinued to build their nest on the previous year's nest if the nest was treated with chemicals, and if it was with enamel paints, something like that. And they discontinue if the colony was burned with fire, and also they discontinue to rebuild their nest if the structure was too old or dead. That means that if that was not strong enough to support the load of the nest. So in these conditions, they discontinue their building the nest even if it was previous year's nesting sites. You can see here the old building before. When it was not so old, they were building, constructing several nests in this building also. Similarly, they were constructing many colonies in these buildings. And this tree, this is the Papachristoforou tree. Now it is dead, but when it was alive, there were more than 100 colonies of Apis dorsata. Now there is no more any colonies here. Similar in this tree also, and in water tank also. Before, when it was very strong enough, there were several colonies of Apis dorsata. And this building, it is new, but they discontinue to rebuild their nest because it was treated with chemicals. That it was whitewashed, it was washed with the enamel paints. Size and shape of the cells. The size and shape of the worker and drawn cells were similar in size. Measuring each size, 0.3 cm long, 1.5 cm deep, and 0.6 cm wide, consisting about 100 cells per 5 cm square. You can see the size of the cells. And the shape of the swarm queen cells was round at the end, measuring about 1.5 mm and thickness being about 18 mm. The thickness of the cell was about 18 mm, and the cell of the honey store were long cylindrical pipes which were very soft and thin. You can see here the cells for the storage of honey and pollen. The maximum size of the honey store was 30 by 50 by 50 cm3 as length, breadth and width. That the maximum size of this honey store. This is the maximum size of the honey store. And you can see the cappings of the queen cells and the remains of the cells at attachment point. The size of this nest varies from 2 to 30 cm in height and 5 to 150 cm in length. You can see this length and this breadth of this comb. The thickness of the comb at the attachment point and bottom in was 7 and 3 cm respectively. That means it was thin at the end, bottom point, and very thick at the attachment point. The maximum size of the honey store, this is the honey store place, was 30 by 15 by 15 cm3 as length, breadth and height. Honey and pollen store was stored always at uppermost corner of the comb, at one corner of the uppermost comb. and brood cells were raised from the thickness, brood cells were raised from the thicker foundation, whereas honey pipes were raised from the thin foundation. You can see this is the foundation for brood production, and this is the foundation for honey stores. Honey is the most. Now, what are the contribution of Dorsata bees to human beings? Honey, as you know, is the most important production from Dorsata bees, and Dorsata bees are highly potential for honey production. Honey from the wild bees, Dorsata is peculiar and might be highly medicinal and highly beneficial for us. Because of the Dorsata bees can forage other than 10 kilometers wider and with a very diversified foraging preference. They can forage farther than 10 kilometers from their nest, and the foraging preference of the bees varies greatly. And pollen and bee bread. They store the pollen as a bee bread at one of the corner of the comb, and 2 to 3 kg of the pollen or bee bread can be harvested in even a single harvest. And the bee bread collected by this Dorsata bees is highly nutritious and may have many medicinal values. And the pollination role of the bees, Dorsata bees, is highly important to increase the crop yield and to enhance the quality also. Now, Dorsata bees in Asia are in danger. Although their contribution for us is very high as honey, pollen and pollination services, but they are in danger because of the loss of nesting sites. That means people, they are cutting trees, people, they are putting fires in the forest, and because of this, our activities, they are in danger. Similarly, on scientific use of pesticides, this is the problem all over the world. We have also the problems with pesticide in Nepal. People, crop growers, they spray pesticide during the flowering period, and because of this, many Dorsata bees, they are dead in the field and on the way to the hive also. And also, we had the problems of pests and predators with these Dorsata bees. We have pests, Theophilidis with Dorsata bees, and there are new predators emerging in Dorsata bees. Eagle is being one of the challenging problems for Dorsata bees in Nepal. And bees, they are in danger because of the unscientific harvest of the honey. You can see here, people, they put the fire, sometimes they burn the colony to get the honey. While harvesting the honey Dorsata, sometimes the honey hunter, they cut the whole comb, including the brood. So because of this, our activities also, we missed many Dorsata colonies. Conclusion, these Apis Dorsata bees, they gave the highest preference to build their nest in man-made buildings, followed by to the trees. And they gave the highest preference to residential buildings rather than to the water tower because of more safe to them from adverse climatic conditions, also more safe from the enemies like birds and other animals. And Apis Dorsata bees shows a highest tendency for the choice of previous year nest. Most oftenly they reoccupy the same spot of the previous years to rebuild their nest. Maybe because of these pheromones of the colony to occupy the same space. And honey and pollen is stored at one of the corner of the comb, and below this they produce the brood. That means the single whole comb is divided for the production of the brood and to store the honey and even to store the pollen. And the nest was tightly attached to the nesting site with a very hard, gritty wax so that it will not fall down easily to the ground. Even following the nest, some parts of the wax remain attached to the nesting sites. The hexagonal shape of the worker and drone brood were similar, whereas the shape of the queen cell was round. The findings can be helpful for the domestication approaches in future. Now, Nepal being one of the richest country in honeybee diversities, but the research work is very poor and we are still in the beginning of the modern beekeeping. So it is very important to conserve these native bees, especially the largest honeybee of the world, that is Apis-Leandros in the higher mountain, and second largest honeybee, Apis dorsata, that we were talking in the sessions, and also another native bee, Apis serena, and another native bee, Apis florea, which are very vital for the point of view of making livelihood, also from the point of making environment better for us. So, it is very important to find some solution to save these honeybees for our kind, for our benefits. Thank you very much.
Thank you very much for this contribution. Are there any questions? Yes, please. Oh, several. Nice. Yes, please. Lady first.
The temper. What is their temper like?
Sorry?
What is their temper like?
The temper.
Temper.
Yeah, they are very aggressive, and we have problem with this aggressiveness. Sometimes they are chased by the eagles, and when they become very aggressive, after the chasing by the birds, they attack any moving objects. Sometimes it can be deadly to human beings or to other animals. Thank you.
Has anybody ever tried keeping these bees in a large bee hive?
Yes, some people they have tried but it's still not successful. I am also trying but it's still not successful.
Do the bees abscond?
Yeah, so the bees they abscond, they leave the hive. Even sometimes they leave the hive, leaving the queen in the hive. Yes.
Any further questions? Oh yes, gentleman over there, please.
These bees migrate. Do you have any records of the migration route and the timing of when they come back to the previous site?
Yeah, that is explained in my full text. Anyway, generally in Chittagong, this is one of the districts of the Nepal, they start to come during the month of October and leave to sometimes up to the month of May, June. But in between this October and June, the coming and going is very common. And rainy seasons, you do not find these bees in Chittagong. They go far away to the western parts where there is less rain and where there is more food available. Yes. Excuse me, can I?
About diseases.
You are asking
Diseases.
about these bees? Yeah, we have. Disease is still unknown about these Dorsata bees, but we have big problem with Termentzi. Because the Dorsata, they are the primary host of this Termentzi. Yeah, we have not seen such diseases still in the Apis dorsata bees.
If I understood correctly, your comment was that if they're tolerant against disease, that would be very nice.
Yes,
Absolutely, yeah.
yeah. They might have tolerance, one. And second point is that they migrate many times in a year, and they become broodless. And because of this broodlessness, they are not much prone to the disease.
Thank you very much for the stimulating discussion, and thank you very much again for contributing. I would kindly ask the technical support staff if there's any opportunity maybe to provide microphones so that we can understand the questions from the audience. That would be great. Excellent, yes. Thank you very much. I would like to ask the audience whether Mrs. or Mr. Reddy is Mrs. or Mr. Reddy in the audience. If that is not the case, then we will have a replacement talk for that one, and Dr. Vincent Dietermann from Switzerland kindly offered to provide such a replacement talk. I think it actually got quite some kindly reminded me on that. I wanted to announce at the break. Because of the very difficult conditions to understand what the people are saying, please get a microphone. So can you hear the English very clearly and we're not disturbed by the noise from the neighboring hall. So maybe if you have problems understanding, get one and you can hear much better. Can wait a couple of minutes. Also interested, please get one and we then carry on, because it doesn't make sense if you don't understand properly what's been said. Yeah.
Carry on.
Good. So Vincent Dietermann, please take over.
All right. Thanks. Well, first of all, thank you for being here, and thank you to the chairs to allow me to fill that blank. And I'd like to present you actually the BeeBook during that presentation. So until now, most of the talks you've been listening to actually were about honeybees. And I'm going to shift the topic a little bit and put it actually on the bee researchers. First, if you read that title, you can read COLOSS. So some of you might not know what COLOSS is, so I'm going to quickly show you in one slide what COLOSS is. COLOSS is a network of scientists that has been founded after the massive colony losses that could be observed in most of the northern hemispheres. So it lasted about 4 years, and after these 4 years, that network grew to about more than 300 members from over 60 countries. And why, within that network, did we decide to produce a BeeBook? First of all, what is the BeeBook? The BeeBook is a practical research manual containing guidelines for direct use by scientists, extension specialists, and beekeepers to standardize the bee research methods. And while doing this, we ended up with more than 1,700 protocols that we selected, standardized for the use of researchers. And each of these methods is really described step by step, so that it's very easy to follow by everyone, even a student that starts working with honeybees. So coming back to the reason, why did we produce such a book? Basically, we felt the need, as every lab started talking to each other to understand the colony losses, we figured out we all do the same things, but slightly differently. So it means that if you do research or ask a question in a different way, basically you cannot compare the results you obtain because you don't know if the differences you observe are due to real biological parameters or are just due to the different method you use to produce these results. So that was our aim. Drosophila, people working with the fruit flies, came against that problem longer ago, so they produced already such standards, and this is what inspired us to do the same for honeybees. For example, on that picture you can see all the different cages that researchers use in the lab to put the bees in and study them. So you can imagine with different cages, maybe different ways to feed them, you will have different results. And this is actually not what we want if you want to compare our results and understand the bees better at the global level. So yeah, our final aim was to understand colony losses. And the drive behind it, I mentioned it already, was the COLOSS network. And we managed to recruit a lot of researchers to take part to this initiative, and we ended up with 230 authors contributing to this method standardization. And we actually went beyond the COLOSS network because only or as much as half of the authors were belonging to COLOSS, and all the rest actually was other bee scientists that did not belong to the network. And we ended up with researchers from 34 countries taking part in the initiative. And actually this is very important for us because what we want at the end is to have a global acceptance of these standards. So we want to involve as many people as possible to take part in the selection of these methods. We also considered each of these different chapters that I will show you about what they contain. We wanted them to be really scientifically sound. So we also recruited reviewers to have a look at all the standards that the authors produced to decide or to prove them and to say, okay, these are really standards that we can use for our work. So as you can see, we included more than 300 people in the selection of these standards, and we hope that it will be globally accepted as a working standard. So what about the content of this book? We have in total 31 chapters. For example, we divided the book, it grew so big at the end we had to divide it in several volumes. So volume number 1 contains all the sort of general methods for bee research, being like you want to study behavior of the honeybee, chemical ecology, molecular biology, physiology and so on, cell culture and colony strengths. And I just want to point out an example on how to estimate strength parameters of a colony because it also has an interest actually in the beekeeping community. In this chapter, the authors actually define how to, for example, define or evaluate the strengths of your colony. And a beekeeper actually might want to do this, for example, if he has to choose the best site to put his apiary, then he can actually measure the strengths and the development of his colony at different sites and then select the better one. Researchers want to do that, maybe beekeepers also want to do that. The second volume is dedicated to honeybee diseases. We start the volume with 2 very general chapters, the one on epidemiology and the one on how to estimate colony losses. If you've been to some other talks of the COLOSS network, you've seen that this is a very important thing for us to be able to evaluate how many bees we lose to see what's going on out there and how big is the problem. All the other chapters of this volume is actually considering each and every known pest or pathogen of the honeybee. And all the methods needed, or hopefully most of the methods needed to actually do research on these pathogens are described step by step in this volume. So this is what we have now. We have 2 volumes and we're already thinking of a third one. Just to give you an example, staying by the second volume, the one I took part to for the write-up is the chapter on Varroa. And this is actually very important for the researchers because we realize how important Varroa is for honeybee diseases, but it also can find use by the beekeepers because in this chapter we explain step by step how to actually evaluate the infestation rate of your colonies. So this can be used also by beekeepers. Now we want to extend further this book by including all the methods needed to do research on honeybee products. So at the end we'll have not 31 but 37 chapters if we consider all the bee products, and we will go beyond these 234 authors we already have. So in this volume we consider all of the honeybee products. And this is at the moment in Khem. So what about the publication? We have different ways to publish this book. We have a hard copy that was printed at the beginning of this month, so it's brand new. And you can see here the 2 volumes, they're quite big books. The first one is about 700 pages, the second one about 300 pages. So there's a lot of material in here. Published here, as I mentioned, this month, and we'll look at the end of 2014, maybe the beginning of 2015, to produce the 3rd volume on the product. So this is for the hard copies, but we also have online versions of this book. Each of the chapters has been published as a reviewed paper in the Journal of Apicultural Research and is available open access. It means you can go to the website of IBRA and download each chapter for free. The first batch of papers was published in January this year, the second one at the same time as we published the hard copy, and the third one will come in 2014 or early 2015. So we have some figures here to show you the interest of the community in this BeeBook in terms of numbers of downloads. You can see on this graph from January onwards, the part 1 of the book was downloaded quite fast and then a bit slower, but still at the end, to this date, we have about 1,200 downloads of the chapters of the first part, and since the chapters of the second part were released, we have about 400 downloads of the second part. So in total, 1,600 downloads, which really represents a minimum of downloads, because then people will send the chapters to each other by email, they will put them on their website, their own website, so then we can't track the numbers anymore, but we know that at least we have 1,600 copies floating out there in the community, and hopefully they will be used for the research. So we have yet another online version of this book, which is also in open access. It's on the COLOSS website actually. We'll link you to the IBRA site for downloads, but the added function we have on the COLOSS website is actually we have a way to make this book interactive. Through a wiki approach, we are planning to give the possibility to the user to make these methods evolve. For example, in some discipline of science, of honeybee research, methods are developed very fast all the time and we need to keep track with them. So we give through this internet platform the opportunity to the user to make this happen, to suggest new methods or to suggest changes to improve the old methods. And this is done this way. Basically you have here a copy of a page with a method described, and at the bottom of methods you can see a little comment field there. This is where the user can put his comments. We'll go have a look at them, and as enough comments have accumulated, we think it's worthwhile to make a new edition of the method, and we'll just do so. We'll include the people that have been given this feedback to write the new versions of the methods. So I told you before, beekeepers actually might have a use of this BeeBook as well. We initially planned it for researchers' use, but beekeepers can find something in there too. So how do you as beekeeper benefit? Basically, this book allows us to have internationally recognized standardized research methods. This will increase the quality and quantity of honeybee research globally. And this is beneficial for the beekeeper because at the end of the line, after our work, actually, our aim is to improve honeybee health and beekeeping in general. So through these better research methods and better research projects, the beekeeper benefits at the end of the line. Because of course, a higher quality bee research will improve honeybee husbandry. This will also, all these methods are also aimed at honeybee parasites and pests. So we'll at the end have better control methods or reach good control methods quicker through the standardization and more people being able to work with each other. So this will end up at the end of the line in better control methods. And there's a lot of beekeepers also in the community that actually like to do research on their own colonies, and the book provides really the methods to do this correctly. If you have a look into the book, the authors also give a lot of background information on the system they're working with, on the kind of research they're doing, on particular biological function of the honeybee, so it's really a mine of information on the honeybee. And volume 3, which will be dedicated to honeybee products, is something, we will have to shout louder now, it's something that can really be used for production by the beekeepers. Again, at the end of the line, these methods will improve honeybee husbandry and production by the beekeeper. And in the chapter that we have, in these 2 volumes of the BeeBook, actually you have, as I mentioned before, methods that can really be used in the everyday beekeeping. Like for example, when we speak about honeybee queen breeding, we also have methods about breeding, about varroa estimation, I told you before. And I think I'm going to end quickly here because I can't really hear myself anymore. We hope really that this BeeBook will be accepted by the scientific and beekeeping community as really working standards, and that everybody at the end produce results that we can capitalize on, that we can compare internationally and really make our work better. So please have a look at the IBRS site where you can actually order the copy print, the hard version, or download the chapters for free. And before finishing, I would like to thank our sponsors. Without the help of the cost at the European level that financed most of the publication of it, this book could not have happened. It's so big and it's not really aimed at making money, it's really a service we provide to the community. Without the help of cost, that would not have been possible. And they've been advertising for the book on their website, so we're grateful for that as well. Ricola Foundation provided also quite a lot of money towards the BeeBook. And of course I have to thank the publisher, IBRA, because it was really a huge task to put together all this contribution for so many people. The webmasters actually who put all the BeeBook on the online platform did also a huge job for this, and I'm grateful to them. And finally, I would like to thank the authors. They produced a huge lot of work to produce this 1700 standardized protocol. And finally, thank you for your attention.
Thank you very much, Vince, for keeping very well the time and not getting too much disturbed by the conditions here. I think, you know, we have a famous saying, it is like it is, except that we cannot change it at the moment, so let's get through this. I can again recommend using the earphones. Even as the person is speaking English, you can much better understand it because you have less problem with the noise. Is there any question to Vince about the topic? Oh yes, please.
You can order it. Because of the logistic problems, they could not bring real books with them, but if you visit the IBRS stand, they have forms where you can actually sign in and order the book. It's stand C43 in the exhibition hall. C43 in the exhibition hall. So just behind you. Is that possible at all? I'm going to check. No, but I can give it to you just now.
Are there any further questions? No, then thank you very much again, Vince. The next contribution will be given by, please excuse me if I'm wrong, Alexandros Papachristoforou. Excellent. Thank you. To afternoon secreted from Honeybee Mandibles during a bite, acts as a local anesthetic in insects and mammals. Please.
Good morning, everyone. She's speaking louder than me, but I'll try. I hope you enjoy the Congress. It's the first Congress where you can listen to 2 parallel symposium. It's amazing. I don't know how they made it. Don't stop laughing. And we're glad to have you here, and also the chairman. I was informed that he was driven to the airport to get kicked out of the country, right? He insisted he's here. Yeah, if you think it's funny, I'll try to make it less funny now and try to compete the lady on the other session. So, I don't know how it starts or from where, but I'll try. Can you see the presentation, please? Okay, we have to wait. It is here. Yes. I'm going to talk to you about a research that started 4 years ago and ended just a few days before I come here. So what you're about to see, most of the results have been published in 3 papers and 1 is on the way. So we're talking about heptanone, secreted by the honeybee mandibles during a bite, acting as a local anesthetic both in insects and mammals. So this is the molecule. It's a very simple molecule, consisted only from 7 carbon atoms, 1 oxygen on the second one, and that makes a very volatile light molecule that is secreted by honeybees and also by ants and other insects, but mainly by honeybees. So, according to literature, during the last 60 years we knew that honeybees secrete this substance from their mandibular glands. The peak, the highest quantity can be found in guards and foragers. Well, there was a dominant hypothesis for many, many years that this substance acts as an alarm pheromone. It means that when a honeybee secretes this against a potential threat, it recruits other guards and other soldiers, as we call them, and they try to attack altogether the potential enemy or intruder. There was another latest hypothesis was that it can act also as a chemical maggot. It means that foragers were placing this substance on flowers that already have been visited, signaling to other foragers that probably it's not worth it to visit again the flower. This hypothesis it's quite has raised a lot of question mark because the compound is extremely volatile. So as I read in some review papers like the one by Breeder Talk concerning the defensive behavior of honeybees, it can be that because it's so volatile that cannot stay longer for a long period on flowers. And concerning the potential pheromone action of this compound for concerning alarm, well, there are many question marks that we're going to see now. The first we wanted to check if it's really an alarm pheromone, if it can act like that. So we applied several doses of this compound at the entrances of colonies, trying to see if they recruit, if it triggers defensive behavior. We had no results at all. The bees didn't care. It didn't cause any reaction. We used also leather targets along with the substance. We spent almost 7 months of continuous experiment to realize that it cannot act as an alarm pheromone. We were not the first to mention this. 2 papers before us, one back in 2010 and one back in 1992, were claiming the same thing. They did experiments with 2-heptanone, and against the dominant hypothesis, it didn't trigger any defensive behavior. So possibly it's not an alarm pheromone. It's definitely there. We know that honeybees use their mandible where they have the 2-heptanone to bite small insects that cannot sting, and there are a lot of references in literature proving this. This is the image of the mandible. You can see tagged with P. It's the pore where heptanone secreted. It goes through a groove mentioned with G, and ends to the sharp mandibles of the workers' mandibles at the sub-edge. We read that it can bees bite waxmoth larvae and varroa. We made some tests with varroa. We don't have any video to see, but we also tried to see this. We tried with waxmoth larvae. This is a slow motion video just to see what happens when a worker detects this, that it's a potential enemy for the colony. It bites, and you can see the response of the waxmoth larvae after the bite at this video. But what we noted after biting was a paralysis of the bitten larvae that was lasting for an average of 7.5 minutes. The larva was there, immobilized, and it was recovering after this period. So this is actually the mode of action of local anesthetics. So we went to to have a closer look, we noticed that all beaten waxworm larvae, small-sized waxworm larvae, had triangular bites at their exoskeleton. So we realized that the bees could actually bite and penetrate the exoskeleton of small-sized waxworm larvae. And probably the 2-heptanon was going in there, the wounds. So we took all these larvae beaten by honeybees and we homogenized them and analyzed them using a GCMS device. And we found that in the hemolymph of all beaten waxworm larvae, heptanon was present. Heptanon was not present in control larvae, of course, and heptanon was not present in recovered larvae. So it means that after recovering, we could not detect any heptanon inside the hemolymph, but it was there in small quantities to all beaten and anesthetized larvae. This is a diagrammatic presentation of what we did. Actually, you can see at sketch A, in figure A, you can see the bite. It's a fixed waxworm larva, and we approach a honeybee worker that bites the larva there. So you see a sense of the potential activity of the larva. It's a motor pattern actually that was lasting for an average of 7 to 9 minutes. And when we injected 2-heptanon, you can see this in diagram B. We had more or less the same effect, though the quantity we injected was 2.5 microliters. It was a huge quantity compared to the one that bees inject in the body through bite. It lasted for around 1 hour and something, and it was more or less the similar like lidocaine. We realized that actually there was something to do with anesthesia, so we wanted a control. We compared the action of 2-heptanon with lidocaine, that it's the world's most dominant local anesthetic now. And we saw that, okay, the result was, of course, stronger and lasted for 6 hours, but the mode of action was more or less the same. So, we realized that honeybees bite enemies too small to sting and inject through the wounds quantities of 2-heptanon that can immobilize the intruder. It's 7 or 9 minutes is enough time just to take it and throw it out of the colony. Actually, honeybees use their own local anesthetic, so that was quite surprising. And since it was acting more or less the same way like lidocaine, we decided to go a step forward, a step ahead. We tested local anesthetic on mammals, actually on nerves and cells, mammalian nerves and cells. We did some in vitro comparison using patch club tests, and we saw that it was blocking the activity of cells. It was the first proof that yes, it can act also on mammalian cell as a local anesthetic. And we ran ex vivo comparison using the mammalian nerve fiber. We actually used the isolated rat sciatic nerve fibers, and we also used the whole sciatic nerve during experiments in the lab, and we also compared other compounds that look like 2-heptanon to see if they have the same effect. So, as I told you, in mammalian cells, it behaved more or less like a local anesthetic, less effective than lidocaine. But this can be excused, this can be explained due to the high volatility of 2-heptanon. Actually, the patch club tests were not designed to test volatile compounds, it was mainly for contact compounds. We skipped that and we went to the ex vivo comparison of lidocaine on the mammalian nerve fiber. We used the champer path, as we call it, the 3-champer path. We actually isolated the sciatic nerve of rats, and we test the compound, and we can watch the compound and take countings every 20 seconds. That was the most interesting result. You can see the nerve compound action potential. On the left, it's when we apply the product. The red line represents 2-heptanon, while the blue line represents lidocaine. You can see that the potential was limited to 0 about 7 minutes after application, and it was more or less equally, though a bit slower compared to lidocaine. You have to keep in mind that 2-heptanone is just a compound. Lidocaine is a fixed product, been used in medicine for many years. Our compound was just in test mode, so but the result was the same with a delay, and the recovery presented actually no differences. So went a step forward again. We tested 2-heptanone with 3-heptanone and 4-heptanone. In all experiments, 2-heptanone was behaving better than all the other heptanones. And we also compare 2-heptanone with other ketones to see, for example, if a ketone with 6 carbons is more effective or less effective. We found that 2-heptanone was the most effective local anesthetic in mammalian cells and nerves. It means that bees have it there, that one and no other, for a specific reason. So we can conclude 2-heptanone appears to play a key role in honeybee defense. Actually, it could be also a good candidate for the development of a local anesthetic to be used in medicine or in vets. It's a natural product, the lipophilicity, it's near to that of lidocaine. It can be metabolized over the same time frame as lidocaine. It's less toxic. Actually, the FDA has evaluated this product as safe and can be a food additive. It's allowed to be added in food for flavors and for human consumption. And this gives us a motivation to start from the bees and go to mammals and hopefully to human. Well, the latest results we have is that a published paper a few months ago showed that this compound can enter the endonebrium 6-fold faster than lidocaine. It's more volatile and can penetrate into the nerves faster than lidocaine. And we also ran some 1-year test to see if there are any analgesic, pain-killing effects. This is the normal behavior used in such experiments. We used male rats of 9 months age. It's mainly for doctor, but the main result was that we found that a higher dose of 2-heptanone, 60 milligrams compared to 20 milligrams of lidocaine, could have the same effect on pain killing. Actually, it can potentially, or in the future, act as a pain killing on mammals. The difference in quantity, it's probably again the same like we found in the ex vivo test. We need more heptanone compared to lidocaine, but always keep in mind this is the compound. It's not a product. We don't even know the correct adjuvants, vehicles to use for the moment. We're making tests, but overall it seems that it's very promising to be used in the future as not only as a local anesthetic, but also as a pain-killing. Probably it will be the first pain-killing compound derived from the honeybees directly for mammals. So I didn't do all this by myself, of course. I had a great team. In alphabetical order is this team with Gerard, Arnold from CNRS, Fokialakis, Kagiava, all these are from Greece, some are from Cyprus, Max Watkins from UK and from Wilde, Europe, who funded this research. And we don't know what will come out in the future, but we're still working on the substance. Thank you for being here this hard morning, especially you to be here, and for the rest of the Congress, of course, and I'm in your disposal for any questions.
Thank you very much. You were perfect in timekeeping. So
thank you. I'm not a part of the organization committee, that's why.
So we have plenty of time for questions. Maybe I will start with the first. Second, I will start with the first comment.
Okay.
Basically, you said that the animal's too small to get stung. So we worked in Africa with smaller beetle larvae. And smaller beetle larvae, the wandering one, they're roughly the same size as wax moth.
It is,
and you can see that when the worker has stung, they change color. They become dark.
We saw exactly the same in wax moth larvae.
They also change the
Yes,
color.
they change the color. They become
white and bluish, something.
Yeah, something like blue-black, yes. I don't know if it's due to heptanone, but it's something that it's very interesting to search in the future.
And I was thinking, like, Maybe that's because what we observed when we were working with the wax mold larvae, they're biting first, and then they put the stinger, so that the local anesthetic may make it more easy maybe for the worker, because the larvae are moving this quickly.
Probably yes. What we noticed, and I didn't mention, is that the size matters here. It's the small waxman larvae were bitten and thrown out. They didn't use the sting. They cannot sting it. It's too small. The larger waxman larvae over the level at 0.01 milligrams at weight, it's more difficult, at least for Apis mellifera, to probably wear the sting. But the case you mentioned now, we don't know it. We've never been to Africa to test this, but it's extremely interesting to test it in the future.
Absolutely. Sorry, there was a question or comment over there from the lady. Yes.
Спасибо за такой хороший доклад. Мы сами очень хорошо занимаемся феромонами.
Unfortunately, the only Russian that I understood is spasiba. I need some headphones to understand the question. Okay, thank you.
А вы оденьте наушник, сейчас вам переведут. Я сама из Уфимского научного центра, очень хорошо занимаемся феромонами. Вот там такое явление есть.
Somebody translate once again because it was dead. Sorry.
Сейчас слышно? Слышно, да? Хорошо. Мы сами занимаемся тоже очень хорошо феромонами. Вы, наверное, знаете наши труды, это Уфимский научный центр. Вот есть там... Ага. Как вы смотрите на такое явление, эффект синергизма? Будете ли вы изучать это 2 гептанона с другими соединениями? Потому что в пчелином улье очень много химических соединений, и там 1-2 гептанон не работает, там работает еще эффект синергизма. Будете ли вы добавлять другие феромоны и будете проводить исследования такие? Будут ли новые исследования, эффект синергизма, будете ли рассматривать, потому что там работает не 1-2 гептанон.
По другим продуктам гипанона, да?
А?
По другим продуктам вы имеете в виду?
Дело в том, что пчелиное улье, там очень много химических соединений, не только 2 гептанона. Поэтому, когда пчела ужаливает, там работают и другие соединения. Будет ли у него новые исследования, добавляя 2 гептанона и смотреть эффект синергизма?
The question for the rest of the people was if we are going to search any other compounds that are present in the colony and not just eptanon for synergistic action or something like that. We're going to investigate. We started some experiments concerning the IPA. It's the second, the most important alarm pheromone of honeybees. Some recent research showing again that it doesn't act as an alarm pheromone. We don't know what it does, but probably we're going to search that in comparison with eptanon. Thank you. Next one.
I guess. Sorry, I think we should carry on with the next presentation. Thank you very much again.
Thank you.
Next presentation will be by Arnold from Turkey. Change level of hygienic behavior in the Caucasian honeybee, Apis mellifera caucasica subspecies. Please, Mr. Güler.
Good morning, ladies and gentlemen. I wait for my presentation. Yeah, thank you very much. I am from Turkey, 19 Mayıs University. The title of presentation is Hygienic Behavior of Caucasian Honeybee and Improving These Characteristics. Again, sorry. This project was financed by Technological and Scientific Research Council of Turkey by TÜBİTAK. And I want to thank TÜBİTAK. Our project team from different faculties, for example, veterinary and agricultural faculty, and also the Honeybee Research Center in the Turkey, Ordu, we studied together. Yeah, and in the last 50 to 60 years, excessive amount of antibiotics and acaricid used against honeybee diseases and parasititis. These chemicals not only caused to decrease the resistance of honeybee, but also caused increase its resistance of pathogens, and in addition, the chemical caused residue in the all honeybee products. However, chemical usage for bee diseases became a common application all over the world by beekeepers. But the colonies die has continued, and for example in Turkey, in each year about 100,000 colonies die from Bacillus larvae, American full brood. In this case, European communities restricted the antibiotics for honeybee diseases in honeybee colonies. The understanding that the chemical use against diseases and pests is not a solution, scientists place emphasis on the hygienic behavior of the honeybee. Therefore, it is accepted that the hygienic behavior is the basic mechanism of resistance to disease and pests. Moreover, this behavior trait can be improved by the selection, breeding programs. Furthermore, for example, Varroa destructor and Bacillus larvae resistance honeybee were improved were breeding by the different universities and research centers. For example, in Minnesota University, by Professor Spivak and her team, they produce Minnesota hygienic honeybee, and on the other side, the Department of Agriculture, United States, produced two different genotypes. One of Russian hygienic bee, the others Varroa sensitive hygienic bees. Yeah, and on the other hand, we have no data. No any knowledge on the Caucasian honeybee hygienic behavior. So we aim to check the variance in the Caucasian honeybee subspecies about this behavior, the hygienic behavior, and this behavior changed or not, and can we improve this behavior or not? Sorry. And here is Anatolia, Turkey. We have about 5 to 6 honeybee subspecies. So Anatolia described as the homeland of honeybee subspecies. The one of them, Caucasian honeybee, on the northeast of Turkey, on the northeast part of Turkey, border of Georgia. This is and Ministry of Food, Agriculture and Animals restricted this area for any genotype, any queen or colony or drones restricted to get in this region. And so the honeybee of this region that we studied about 25 years, this region honeybee Caucasian, and it is pure. So in the 2010 we bought 400 queens from queen rearing enterprises from this region, and we carried them on the Ordu and 19 Mayıs University Samsun into institution for protect the colonies. Yeah, at the beginning, all trades, all management were equalized in the colonies at the beginning, in the 2010s. The colony that swarm, lost queen, diseases, and different morphological characters, we remove them from the study. So in the first generation, from 400 colonies, only 167 colony finished the experiment. And also we measured that pupa removal, adult worker bee population, brood production, honey yield, box production, varroa destructor infestation, and standard 41 morphological characters. And all colonies' performance also determined. And this is our standard distribution of honeybee subspecies in the Turkey. This, especially our main subspecies Anatolia, Apis mellifera anatolica, and the others. Two types we cannot. They maybe we say ecotypes or Caucasian in here. Yeah, this is standard. This shape doesn't change when you or anybody use the morphological standard. The colony that in our study, if they don't suit this distribution, we remove them from study. And in the first generation, 309 queens were instrumentally inseminated, and in the second generation, 293 queens were instrumentally inseminated. And from each selected colonies, each queen represents by her 10 to 12 daughters. And also each queen inseminated with the semen or spermatozoa 35 father colonies, except her father and mother colonies, each queen. That's the application of liquid nitrogen for determine the hygienic behavior. And we applied 165 pupae, about 300 cc or milliliter of liquid nitrogen, and applied to 165 pupae. Yeah, that's the results of hygienic behavior. The cleaning pupa by each colony in the first and the second generation. In the first column, in the first generation in here, and the highest colony cleaned 99.6% of pupa, and we select the colonies cleaned 95 and over %. But in the second generation, there isn't any colony that cleaned 100%. But in the second generation, 19 colonies cleaned 100% of that pupa. And the results of our study, I combined. That's the results of the first generation from 400 queens, Yeah, that's the average dead pupa, minimum, maximum, and 36 colonies were selected. They had hygienic behavior. In the second, that this colony is the mother or the father of this generation, and so these are the parents of third generation, mother and father of this generation. Yeah, and in the first generation, only 36 colonies were hygienic, but in the second generation, it is increased to 160 colonies. And in the 2013, we raped and instrumentally inseminated 278 colonies, and now we applied liquid nitrogen, and our study continued. So I want to say something. In the first and the second generations, only we measured selection index. In here, as seen, yeah, the number pupa differentiation between the first and the second generation, and the selection index, yeah, in here, it is about, yeah, we can say something, and this trait can be improved, as now we are aware of that. And the conclusion, the change in hygienic behavior in two generations might have resulted from the genotype of these subspecies. For that reason, it was thought that the hygienic behavior of these subspecies can be improved. Study will be continue until the end of 5th generation. We will continue the end of 5th generation, and then we will measure the selection index and the coefficient of heredity and the breeding value of all colonies. We will measure it. Yeah, and thank you very much for your attention. Thank you very much.
Thank you very much. You've been perfectly well. We have plenty of time for questions. Over 7 minutes. Is there any question, please? Yeah, over there, the lady was first. The back.
Where you have so many colonies together in your mountain district, in your valley, mountain valley, is there enough forage for all those bees? And what forage do they have?
Sorry, please could you repeat your question?
Where you have so many colonies together in the mountain valley, is there enough food for the bees to get?
Yeah.
And what food is it?
Yeah, thank you very much. That we divide 400 colonies into two sides, 200 colonies in each institution. One in the university apiary, the other in the research center. And we applied migratory beekeeping. Yeah, from Black Sea to East Anatolia. About 600 kilometers, there are a rich area for flower, young for flower, different. Yeah, for the rich region. Yeah.
Yes, please.
Just let me know your question. I'm going to tell it. What you want to know, please. Varroarification of the bee. It's bee. It's a bee.
The question is whether you talk about the generation of the varroa mite or the generation of the bee.
our generation of 4B, not varroa. We measured the varroa sensitive in each colony. We measured it, but I didn't give any data. Only my data for hygienic behavior, the colony behavior. That's okay?
No, I'm saying because you're harmonizing the two.
Sorry, yeah.
So you're considering the hygienic behavior in terms of the varroa and the bee. So how are you going to know that the bees are removing the varroa mites? How do you harmonize the hygienic behavior?
Yeah, thank you. I understand your question. That we don't know, not certainly. That's in this stage we cannot say anything. We will wait the end of study. But we know that if the colony cleaned 95% of pupae, it should be hygienic against American full-brood and varroa. Yeah, in the study we know that, and the improving genotypes, they track the hygienic behavior and the relation between hygienic behavior and the varroa cleaning.
Okay, thank you very much again for your contribution.
So, sir, there was one more question from
There's one more question, please.
Yes.
Sorry.
Sorry, wait. There was one more question from the person. Short of time? Thank you. No. How did this breed affect the...
No, thank you. We are running late. I would like to thank very much all the contributors and the audience for their patience. And we would like to carry on with the next talk will not happen. So we have a break now, but I would like to very much use the time now for the local technical team that they may coordinate with the other room. So please come back here 20 minutes past 11 after the coffee break and we're carrying on the session. Thank you very much, all of you. 20 past 11. I was told.
Yes.
Hello. Thank you. There was a misunderstanding there, and here I will give my presentation on ethyl-8, the biosynthesis of ethyl-8 and the seasonal variation in the honeybee apis Everybody knows honeybee has a very complicated way of communicate. One of those is through pheromones and hormones. Pheromones are produced for different members of the colony, even the larva, the queen, and the workers itself. We don't know exactly if drones produce pheromones or not, but that is not today's presentation. And we see from the eclosion and the different larval modes of the honeybee, there is an interaction between hormones. We see ecdysteroids and juvenile hormone, peaks and lows that produce these changes towards the adult honeybee. And once the adult emerge, there is a steady increase of juvenile hormones that produce the changes in behavior of the honeybee from inside the hive, where they carry the brood, towards when they age to foraging that is outside the hive for food. This ratio between bees that go outside the hive and the bees who are inside the hive is very important for the homeostasis of the colony. So they have a very good number of bees bringing food into the colony, but at the same time have enough bees to care for the brood. And this transition is influenced by the production of pheromones. One of these pheromones is beta-oxymine, is produced by the brood, that is faster the transition, and the other is, for example, produced by the foragers, is the ethylolate that slows the transition. For this experiment, we use cohort colonies, so we have known numbers of bees with different age in a colony, and we can detect how many days it takes for a bee to move from inside the hive to outside for foraging. And this treatment was the effect of ethylolate, so these colonies were treated with ethylolate in a candy to see the effect on the number of days for foraging. We are interested in ethylolate, and this experiment, there is several experiments, many figures. Because of the short time, I will point out only the very specific results. This is our hypothesis. Ethylolate is an ester formed from oleic acid plus ethanol, and can be produced internally in the cells by membrane of coenzyme A, and also an esterification between oleic acid and ethanol by alpha-beta hydrolases. But this enzyme, the alpha-beta hydrolases, have also two ways. They produce the ethylolate, but at the same time they destroy by hydrolysis the ethylolate. So we were interested in three questions. What are the sources of the precursors of ethylolate? What is producing the ethylolate in the honeybee? And also, what is the seasonal variation of ethylolate during the season of the honeybee? We did tissue perfusion experiment, so we sectioned the honeybee into the three-part head, thorax, and abdomen, and we add the substrate that has a deuterium label. In this way, we can track the production of ethylolate. There is many ways to read this, but one simple way is that when we add the substrate to the head, to the thorax, and to the abdomen, we found consistently that the part from the forager produce more ethylolate than the part from the nurses. And most of the ethylolate is produced in the head. So now the question is, this is in the laboratory, but in nature, where does come the ethanol from? Well, the ethanol comes from the nectar. There is huge amount of yeast, natural yeast, wild yeast in the flower. So this yeast has the capacity to produce nectar from sugars. And this sugar is taken by the honeybee, and they are esterified with the oleic acid that are in their body and produce the ethylolate. Our experiment used two labeled substrates. One is the D6, or 6-deuterium ethanol, and oleic acid with 4-deuterium label. This is one of the feeding and vapor experiments that we did. So we put bees in a cage and we feed syrup with labeled substrate. Between the several analyses, we found, for example, that the amount of labeled ethylolate in the honeycomb of foragers that were fed directly with this labeled substrate, and those that were exposed only to the vapors of the ethanol, produce almost the same amount of ethylolate. But that's not the case in nurses. When the nurses were exposed and feeded nurses produce much more ethylolate than nurses exposed only to the vapor. And in the case of we put in two cages with one nurses, one foragers, and the ethylolate, the serum with the label ethylolate was in the middle, were not fed directly, they were only exposed to the vapor. In both cases, we found that the nurses and the foragers exposed to vapors of ethanol, diseased ethanol produced ethylolate. But when we feed the nurses and the forager with denied ethylolate, we couldn't find any traces of ethylolate in the honey crop. We only found in the exoskeleton. So there is produced inside the body, but it looks like the bees exuded the ethylolate and through the cuticle, and that's where they produce the pheromone effect. How to identify which genes are responsible for this esterification? Well, we did an in silico analysis in computer for several families of genes that are related to this enzymatic reaction from literature in other species. And then we target this gene with the generator primers, we clone these amplicons, and we identify by sequencing which are these genes, and we clone this gene, and we did a quantitative expression analysis of these several genes, and then in situ hybridization, in a specific gene we clone and we express the protein, and we did in vitro activity assays. On the several genes we test, we focus on 4 genes, the alpha-beta-3 gene, long-chain acid-CoA synthase, fatty acid desaturase, and fatty acid transport protein. What is important is, for example, in all the genes, the fatty acid desaturase was the only where there was difference between 1-day bees that were not treated with 15-day and 15-day all treated with EO from the original cohort hive. And the fatty acid desaturase was the only one which showed difference when we evaluate 15 days versus 15 days all treated with EO. So now is where is ethylolate biosynthesized in the bee? We did the in situ hybridization, and we consistently found that in this head section, we found that this gene expressed in the esophagus of the honeybee. The high expression of the alpha-beta hydrolase 3 and the fatty acid desaturase and the fatty acid transport protein, but very low expression of long-chain acetyl-CoA synthase. We did another section, the thorax section, and we can see the esophagus with high expression of this gene. And when we see a close picture, we see that this gene, alpha-beta-3 gene, expressed in the internal epithelium of the esophagus. So then we try for in situ activity. We feed the honeybee with 2-nastylolate, ethanol, and hydroxy peroxidase and whole-radius peroxidase to check the enzymatic activity in general of esterification in honeybees. This is the reaction that happens when we feed with 2-nastylolate. Is converted to 2-nastol with the ester synthase, and the 2-nastol it could be converted into a fluorescent dye. So this is the section we found again that the fluorescence appeared in the esophagus of the honeybee. So this confirmed that there is esterification in the esophagus of the honeybee. And the honeycomb has very strong signal of esterification. So the ester synthase, this esterification that we saw in those pictures, are the product of the alpha-beta 3 gene. So we need to connect these two results. So we think that EO, the ethyl oleic, is produced in the honeycomb and it's due to the exoskeleton. The second result is messenger RNA is detected in the esophagus. Trans esterification, the enzymatic reaction occurs in the esophagus. And when we see the alpha-beta gene, we see that it has a signal peptide. So theoretically it's released into the regurgitate or into the crop fluid that is in the esophagus. And one important thing is many studies have found that there is no EO, no ethyl oleic in the regurgitate or in the crop fluid. So crop fluid looks interesting. So we did an SDS page analysis of the crop fluid or regurgitate, and we found several proteins, but no one specific protein was interested. So what we did, we do separation of different proteins by fast protein liquid chromatography, and the different fractions were analyzed by activity. We cultivate this fraction of the regurgitate with D6 ethanol and D4 oleic acid, and we found that it has activity. So the regurgitate or crop fluid has enough enzymes to produce EO. And these were confirmed in our experiment. Not all the fraction produce EO, but there was one important peak around fraction 23-24 that produced EO. And also the regurgitate produced large amounts of EO. So we checked for peptides in the regurgitate, and we found several proteins, but one interesting protein was the esterase lipase, that is another alpha-beta 3, and also other binding proteins that I will talk a little bit later. So we express the alpha-beta 3s and the esterase lipase in Pichia pastoris, and we get almost single band proteins, and we analyze the activity by enzymatic kinetic analysis, just to show that it has the properties of an enzyme. The in vitro activity showed that when we cultivate this protein with the ethanol and with the oleic acid, we produce ethyl oleic. But at the same time, these proteins, alpha-beta 3 and esterase lipase, have hydrolysis activity. So in one way produce EO, and the other way is destroy the EO, hydrolysis EO. So what we need is, there should be another protein that attached to EO to hindering from this enzyme. So we use BS8. We know BS8 is a protein that attached to EO, and when we did the cultivation with this enzyme plus the BS8, we found that in presence of BS8, the production of EO is higher than without BS8. And when we did the hydrolysis analysis, when there is no BS8, the hydrolysis is high, but when there is BS8 in the reaction, there is almost no hydrolysis. So BS8 is saving, is protecting the EO from hydrolysis. So we did another in vitro activity recombinant protein to know this enzyme, what is the preference, and we found that in the case of in vivo, there is a preference for C13 carbons, very high preference, for ethanol alcohol. But in vitro, the preference is a little bit different. There is a preference for oleic acid and for 1-propanol in the case of alcohol. So after that, we detected this enzyme are responsible for EO. then we ask, how is the production of EO in life? So we test by the season, and we found that foragers that collect nectar produce more EO than foragers that collect pollen, and much more EO than nurses. So we did an analysis during the season, and we found some peaks during the summer, in August, and in the other year around July. So this is the reaction that followed when we did in-cage experiment. So we feed with label substrate, and we produce D9EO when they take the label oleic acid and the label ethanol. And in winter bees, we found that when we feed bees with label substrate, there is a high production of EO at 4 hours and then decrease clearly afterward. And most of the production is in the exoskeleton. You can see there is a huge increase in the exoskeleton and almost a base, no changes in the honeycomb. And the second experiment is we feed for 4 hours, then we change the feed to only syrup without any substrate, and we check the production of EO. So in zero mean, after 4 hours of feeding with label substrate, we have a high amount of EO in the exoskeleton, and this amount keep constant around 2 hours and start decreasing after 4 hours. But the EO in the honeycomb decrease right away and keep constant. So it looks like there is an accumulation of EO in the honeycomb, and over that level then start exuding the EO. So there is, we need to see how is the interference of juvenile hormone 3 in this production. So in this case we use methylprin. We see here that when we add methylprin in the equation, there is a higher production of EO into the exoskeleton. So it looks like methylprin, what it does is increase the exudation of this hormone. And there is a more complicated figure here because we use summer bees separated between nurses and forager. But to be simple, there is a high production of EO in the honeycomb and the exoskeleton when we feed foragers with label ethanol and methylprin than in nurses. And then we see the effect of EO, methylprin in the honeycomb, in the fluid in the regurgitate alone, and we found that the regurgitate from bees treated with methylprin produced more this NIEO. And at the end, we check the seasonal variation of the O production in the regurgitate. We take regurgitate from May to November, and we check how is the activity in vitro. So we found that the regurgitate for May has a very strong capacity to produce EO versus compared to the other months, and almost disappeared during the late fall, early winter. So this is the biosynthesis of EO in the esophagus and the honeycomb accumulate, and then there is a rapid and quickly accumulation in the honeycomb and then exudation to the cuticle. And that's where it produces the primary effect. So it looks like the bees need to be at least 19-20 days old to be able to take the ethanol and produce the ethyl oleic. So there is only 19 days old at least to take ethanol and produce EO. And this is more or less what we think about this confluence of different hormones and pheromones in the brood area and the estuarine area and outside. So as the bee age and change position, they are more exposed to ethanol from coming resources, and they produce more EO. So the higher number of foragers produce more EO, and so they delay the transition from nurses to foragers. So the conclusion is that both nurses and foragers can synthesize EO. The second is we have two genes that produce EO in vitro, and they express in the esophagus, where is the location of EO production, and they accumulate in honeycomb, and above level they are exuded into the cuticle. And that the metroprim, what it does is increase the exudation, increase the presence of EO in the colony. And this EO follows the growing season of the colony, and this is the product of non-exudative metabolism of ethanol. So EO, what it does is sensing by the other bees by antenna and by olfaction, but not by tropolaxis. There is no interchange of nectar, there is no EO in the nectar or in the remodite. And that's the way it produces the primary effect. So a lot of foragers produce a lot of EO, and the EO, what it does is delay the nurse to forager transition. If something happens with the forager, there is a storm of the population of foragers down, so there is low EO in the colony and the transition go faster. So nurses became foragers faster than the other. So I want to thank the NSERC and the Human Frontier for their contribution to the program and all the collaboration that you see there. Thank you, and I think I pass for a couple of minutes. Thank you.
Thank you very much. I'm actually happy that the majority of the people currently stayed in the room and that we had an opportunity to listen to this interesting talk. And there's time for one question. All right, then thank you very much again. I believe that we have now